international lecture on Brain Talk, Engineering Solution to Communicate with the Brain, organized by Department of Chemical Engineering, St. Joseph College of Engineering, Chennai. I specially want to welcome our guest, Dr. Jitendran Muthuswamy, working as an Associate Professor in Department of Biomedical Engineering at School of Biological and Health System Engineering, Arizona State University, USA. To start with, human brain is a command center for human nervous system. It receives a signal from the body's sensory organs and output information to the muscles. The human brain has the same basic structure as other mammal brain, but it is larger in relation to the body size than any other brain. The study of anatomy of brain is called as neuroanatomy, while the study of this function is called as neuroscience. Numerous techniques are used to study the brain. The specimen from other animal, which may be examined microscopically, have traditionally provided much information. Medical imaging techniques such as functional neuroimaging, EEG recordings are important in studying the brain. Brain research has evolved over the time with philosophical, experimental, and theoretical phases. Our guest will explain about the various biomedical engineering solutions to communicate with the brain. Now, I will hand over this session to our guest, Dr. Jitendran Muthuswamy.
Thank you very much, Karthikeyan, for the introduction. And thank you, Bhaskar, for your kind invite to come and talk to your students and faculty of the St. Joseph's College of Engineering, Chennai. It's a pleasure and a privilege and an honor to talk to you all today. The topic of my presentation today will show a slice of what we do in our lab over the last 15 years of research at Arizona State University. I just want to clarify the title first, Brain Talk. When I use the word brain, I use it in the sense of its physical nature, with its constituents, neurons and glia and the vasculature, and its extracellular tissue, and its function, and all of its functionality and complexity. We're not talking about the mind, but we're really talking about the brain. And indeed, that's a subject of fairly intense debate as to what is the mind and what is the brain. And we're not going to get into that. But I just want to point in that direction to students who might be interested in reading more about it. The mind versus brain debate has been going on for a long time now. I'll read a couple of quotes. The first quote is from Francis Crick, whom you probably all know as one of those. James Watson and Francis Crick were the two people who discovered the DNA structure, the DNA molecule. So this is a quote from Francis Crick. You, your joys and your sorrows, your memories and ambitions, your sense of personal identity and free will are in fact no more than the behavior of a vast assembly of nerve cells and their associated molecules. So that's one perspective. The counter perspective is from John Polkinghorne. He's a quantum physicist and a theologian as well. He has an interesting perspective. He says, if Crick's thesis is true, we could never know it. For not only does it relegate our experiences of beauty, moral obligation, and religious encounter to the epiphenomenal scrap heap, it also destroys rationality. Thought is replaced by electrochemical neural events. Two such events cannot confront each other in a rational discourse. They are neither right nor wrong. They simply happen. The very assertions of the reductionist himself are nothing but blips in the neural network of his brain. The world of rational discourse dissolves into the absurd chatter of firing synapses. Quite frankly, that cannot be right, and none of us believes it to be so. So very interesting. So the reductionist perspective of Francis Crick doesn't appear to be sustainable from a philosophical point of view. But leave that aside, we're going to focus on the physicality of the brain. How do we communicate with the brain? And much more mundane aspects of why do we even need to be communicating with the brain? And how do we do it and all that? So the outline of my talk is why communicate with the brain? How do we communicate with the brain? What is the science of neural implants? So I'll talk a little bit about the science before we dive into the engineering aspects of this talk. There are a lot of technical challenges associated with this, which I'll highlight along the way. I'll talk a little bit about the technologies for monitoring and stimulating the brain. So this is a two-way communication. Monitoring is like reading the brain, and stimulating is like writing into the brain. And I'll talk a little bit about three distinct approaches our lab has pursued over the last 15 years, and I'll highlight some of those key contributions along the way. And then I'll end with a couple of thoughts on having a successful engineering career. All right, so complexity of neural networks. This is a picture which shows about 302 neurons in a worm called C. elegans. What you see here on the top left is all of the neurons. It's a complete wiring diagram. And what you see in the bottom left is your connectivity pattern. It shows every single neuron connected to every other neuron. And it's been painstakingly done over with a lot of effort and from a lot of different groups. Again, the goal is to find out, what does this all mean? How does this all work? Now that we've found out the functionality, the diagram, the circuit diagram, the difficult part remains as to find out how do we determine the function. So you can imagine the complexity of the human brain with its billions of neurons. There's a huge effort in the United States and across the world. Billions of dollars are being poured in to figure out the circuit diagram of the brain. And we're making tremendous progress. At the end of the whole thing, we might have something that looks like what you see in the top left, except that the 302 neurons will be replaced by billions of neurons, and all the connectivity and all its glory will be there. And we will be left scratching our heads for trying to figure out, what does this all mean? And how does this all work? If I have to move my arm, what circuits are being activated and what circuits help me to achieve that purpose? Ultimately, that's what we're really interested in, functionality of the brain. So as we're going to talk about the functionality of the brain, I'll talk very briefly about some of the signals that you can actually get from the brain. And the reason I talk about this electrical signals here is because they're central to every investigation you launch into the brain. Every hypothesis you set up and every experiment you test, you use to test those hypotheses, eventually at some level will rely on signals like this to test if the hypothesis is true or not. So all of our knowledge of brain function and dysfunction, much of it is obtained by signals such as these. So I'll just slowly go over some of these signals here, starting from the bottom right. What you're seeing there is if you were to take a wire, a conductive wire, and stick it inside a neuron, a cell body of a neuron. And if you're a good engineer and you design the wire sufficiently with a sufficient impedance and exposure at the tip and all that, you would see a signal that looks a lot like what you would see in the bottom right corner, with sharp spikes and a DC or constant voltage. Your reference electrode is outside the cell, your electrode is inside the cell. These spikes are what the cell is, the cell's information. It tells a lot about how the cell communicates with the next cell that it's in contact with. It tells us a lot about how does it encode information. If there is an event outside, if it's a cell that's responsible for vision, if there is a visual event, how does that visual event be, how is that represented in this cell? Is encoded in those such activity. Now, if you pull that wire a little bit outside the cell, you'll see something that looks like the trace that you see here. Very noisy. You still see those spikes, which are called the action potentials, but you no longer see those constant DC potential, but instead you see something that's very low voltages. These voltages are typically in the order of a few tens of microvolts to a few hundreds of microvolts. We call them extracellular action potentials, EAPs. And let's say you move the electrode further out and you appropriately design it, so you're actually listening in not just to a single cell, but you're listening into, let's say, hundreds of cells. You would see something that looks like a lot like this trace that you see here on the top right, which we call LFPs, which stand for local field potentials. And the amplitudes of these are typically in the order of hundreds of microvolts to maybe in the low millivolts. And you go down here further to the bottom left. If you had multiple electrodes implanted in different regions, you'll see multiple traces, multi-channel local field potential activity. And if you had electrodes sitting on top of the brain, on the cortical surface, we call it the electrocorticogram, and that looks a lot like this trace. Very similar to local field potentials, but it's an average, an ensemble average of thousands of cells, thousands of neurons. And then you go up a little bit to the last trace EEGs, which some of you might be familiar with if you've been to a hospital and done sleep studies. This is where they put the electrodes on your scalp. They don't put it on your brain, but it's on your scalp. And that picks up events or activity that's volume conducted electrical activity that's being conducted through the brain, through the skull, and onto your scalp. And it's an average of all kinds of, about thousands of neurons that get propagated and volume conducted through the brain. So that's your electroencephalogram, or EEGs. So that, in a nutshell, gives you all the different types of electrical waveforms you might see in a brain. They're all from different scales, from single cells all the way to hundreds of cells, to thousands of cells, to EEGs, which actually pick up activity on the scalp over thousands of cells. So this is what we have at our disposal in order to be able to understand the brain. All right. So this picture here, where I put actually all the brain monitoring techniques that we currently use in one picture, with the x-axis being spatial resolution and the y-axis being temporal resolution. So just look at this carefully. The spatial resolution is highest on the bottom right, starting from 1 micron. A micron is a thousandth of a millimeter. And if you go along the x-axis all the way from the bottom right to the left, you come to centimeters, or 10 centimeters. Now, if you go along the y-axis, you go from 100 seconds at the bottom all the way down to 10 microseconds at the top. So the highest spatial resolution is at the bottom right. The highest temporal resolution is at the top left. All right. So you map all these techniques that we have right now, and our ability to read events in the brain is mapped into this in a spatial temporal resolution. So the highest spatial temporal resolution is probably the patch clamp right now, where you actually have cells, individual cells, and you're actually measuring events inside the cell. You're picking up membrane potentials, and you're picking up ion channel activity across protein channels in the transmembrane proteins. So you're picking up ion flow, ion flux in ion channels in the transmembrane. So you're looking at conductivity of these ion channels, you're looking at membrane potential changes, all in a single cell. So that gives us the highest spatial resolution and temporal resolution. And the next up is extracellular action potentials. And then slowly you move on down here to ECOGs that we saw before, EEGs, and then fMRI that you alluded to in your introduction, and the positron emission tomography and all that. You get down to really low spatial and temporal resolution as well. So if you want to have, so what is our goal here? What is the ideality? I'll lay the ideality down here. We are nowhere close to achieving that, but if you really want to understand the brain, the holy grail, if you were to ask somebody, what's your dream wish or dream technology that you would like to have, if you ask a neuroscientist, he would probably say, boy, I wish I could monitor every single cell in the human brain. I wish I could monitor the electrical activity of every single cell. I wish I could monitor those cells as long as possible. And the third wish he would say is, I wish I could monitor them as long as possible while the person is behaving. Not under anesthetized state, but under behavior. So you can think of that as the 3 legs of a stool. You want to monitor as many neurons as possible, as long as possible, in an awake behaving person. Why awake behaving person? Remember, the goal here is to find out how is behavior encoded in the activity of the neurons. So that's how we find out the function of the brain. We want to know, how does the brain accomplish its function through these kinds of signals and so on. So those are the 3 desired goals anyway. So let's move on. So the purpose here, we address the question of what are the different ways you can monitor the brain. Why do we even want to do this? Why do we want to read the brain? Why do we want to write into the brain? So spend a few minutes here talking about the different reasons why we want to be monitoring the brain. So there are a lot of prosthetic technologies that are already out there, huge success stories and emerging prosthetic technologies. I will start with the cochlear prosthesis, which is a huge success. And that's really driving a lot of these other technologies coming forward. The cochlear prosthesis now enables people, and generally the way this works is you have a person who is profoundly deaf have these things implanted in his ear canal, and there are electrodes in these ear canals that actually go in and stimulate specific auditory nerves. So the point here is that the person is unable to hear. You use a microphone to capture the voice or the sound from the external world. You have a sound processor that then converts this external voice or whatever signal he wants to listen to. It's converted to a stimulation patterns that is then wired down, that is sent down through these electrodes that go and stimulate appropriate nerves in the ear. And this is so successful that profoundly deaf people can have a telephone conversation these days. So that's a huge success. The next step that's coming up is a visual prosthesis, which has been, there's a company called Second Sight in Los Angeles, and there's several companies across the world that are actually actively working on this. Electrodes implanted in the ear, actually stimulating the ganglion cells in the eye. The point here again is specifically targeted right now towards patients with retinitis pigmentosa, in patients in whom the photoreceptors are degenerated. So these electrodes replace the function of those degenerated photoreceptors. So they get the light. The light is then, the camera captures the scenery outside the patient. That scene is then converted to a bunch of electrical signals that are then sent to specific electrodes in a predetermined fashion and stimulates specific cells in the retina. And the patient is not necessarily able to see the scene, but the patient can right now figure out contrasts, black and white, colored, and so on. They can actually see the frames of a door. So I've heard people give these testimonies in conferences. They can see the frame of a door, so they can actually walk through the door. So it's not full vision, but it's getting there. And people really appreciate that. I talk to the patient, it makes a huge difference to them right now. And to the lower bottom left, you have cortical prosthesis, patients with electrodes implanted in the motor cortex of the brain. They can actually use that to control an external robotic arm, such as what you see here on the bottom right. Those are some examples of current prosthetic applications. Other applications coming up include, actually, some of them are already there. For prosthetic devices for neurological disorders such as Parkinson's disease, and these procedures might be are routinely done. This was FDA approved in 1996, where electrodes are implanted in the subthalamic nuclei and different parts of the basal ganglia circuit. Parkinson's disease patients have a tremor associated with them. So this has been a remarkable success that electrodes implanted in the subthalamic nucleus, when stimulated with appropriate dose of electrical current, can actually completely mitigate the tremor. So people can have normal lives. This is a huge success. And that's driving some other investigative pathways here, where people are looking at the possibility of deep brain stimulating therapies for Alzheimer's disease and even mental health disorders, neuropsychiatric disorders such as OCD, obsessive compulsive disorders, treatment-resistant depression, and a variety of other disorders. People are exploring the possibility of using deep brain stimulation to look at these things. Memory and cognitive enhancement and restoration is another interesting application. That's kind of looks like this, where you have, this is a picture in purple, is the hippocampal circuitry, which gets afferent signals from the dentate gyrus. And then it's the CA3 gets that afference from dentate gyrus that's then routed to CA1. Let's say you had a stroke and you had an infarct. So this red cross mark represents a damaged part of the hippocampus. So that is no longer functional. You could implant a chip here in the brain that receives the signal that should be going to CA1, but now it's not going because of this infarct. That signal gets routed to the chip, and then the chip then processes it and will give out a signal exactly the way CA3 would have given the signal. So the brain actually doesn't know if it's actually from CA3 or is actually coming from the chip. If you've done it that well, you're doing it. You have achieved success. And there's a tremendous amount of progress in these kinds of chips, which actually get these inputs and are able to receive downstream neurons and restore memory. Hippocampus is known as a structure that's responsible for memory and cognition and so on. So that's a work in progress, which has gone from Ted Berger's lab in University of Southern California. This is a picture of what's out there so far. I talked about the deep brain stimulation implants for Parkinson's disease, cochlear implants. I've already talked about that. Vagal nerve stimulators, again, a huge success story. You can actually implant these things in patients. The procedure, I timed it, actually takes about 20 minutes for the experienced surgeons to implant these things. Once the patient is all set up, he cuts open the patient in 20 minutes. He gets this cuff electrodes wrapping the vagus nerve and stimulating with an appropriate dose can actually eliminate epilepsy, seizures, and so on. Control of artificial limbs, I talked about that as well. Sacral nerve stimulators for controlling bladder function, spinal cord stimulation for pain, retinal implants for restoration of vision, cortical electrodes for brain mapping, and also for a readout. It's good to have a cortical readout for a variety of reasons to know what exactly is going on in the brain in response to your stimulation. So there's a lot of, all these things are already out there in the clinic. And many of these are actually in the clinic. So there's this idea of bioelectronic medicine. I just want to put it out there. This is another reason why we want to be reading into the reading signals from the brain and communicating with the brain. Where the idea here is, instead of the existing paradigm of molecular medicine, we are actually designing molecules that target specific receptors that then to address specific disease states or end organ dysfunctions. What you're doing here is something radically different. You implant sensors that measure the physiological state of the person. The sensors can be anywhere. Preferably here, in this case, you're reading data from a neuron or a set of neurons that somehow indicate the functionality of the end organ that you're interested in. And that signal is taken out, and you assess the physiological state of the end organ, make an assessment, is it really normal or not normal? And based on your assessment of the target state, then you generate a controls algorithm that generates a stimulation signal. And that stimulation signal is then routed back into the nervous system at a different location, and you stimulate it. So for instance, the vagus nerve is actively being investigated for a variety of disease states, inflammation, asthma, even hemorrhage, obesity, diabetes. So a lot of disease states, there's over 10 different disease states where this type of paradigm is being investigated. So those are all the reasons why you want to be reading the brain, and there's many more. And those are all the reasons also why you want to be communicating with the brain, why you want to be stimulating the brain as well. So how do we do this? So let me launch into the how question. We've been doing this for decades now. We've been reading data from the brain at all those different scales that I mentioned earlier: intracellular, extracellular, local field potentials, ECOGs, electrocorticograms, and EEGs. Across different scales, we've been recording for several decades now. How are we doing this? We do this by using technologies such as what I'm showing you on the slide. It looks very crude, but it's just a piece of wire. You can actually put these wires, when they're designed the right way and put in the right place, you can listen to single neurons. So in this picture in the left, these triangle-shaped pictures are in blue are pyramidal neurons in the cortex. So they look like a pyramid, so that's why we call them a pyramidal neuron. They're pretty large, and they extend for several millimeters. The processes extend for several millimeters. The cell body itself is pretty tiny. The scales are written down here, and it's fairly accurate. It's called 50 microns here. Again, a micron is 1,000th of a millimeter. So you're really trying to listen to a cell that's very, very tiny. And when you're close to the cell body, you get the largest signal in terms of the action potential that you can pick up outside the cell. So by default, now I'm assuming that we're going to go into the extracellular action potentials. Again, the reason is because you may ask, why not go for imaging? Why not go for the other techniques that I put up there, or EEGs or ECOGs? The problem with them is the poor spatial and temporal resolution. So if the goal really, remember, is to monitor all the cells all the time, in awake behaving patients, we want to be able to get to single cell level. So that's our ideality. So we want to be able to put wires at every single cell. Again, that's not possible right now, but I just want to put that up as an ideal goal. So you put the wires right next to the cells, and you're monitoring function. And the wires look like this when you take a cross section. Back in the 70s and 80s, with the emerging advances in silicon technology, people started asking, okay, if we want to be measuring activity from thousands of neurons, we can't be putting one wire at a time. That's too tedious, and it's impossible. It's going to take a long time. Why not just use this batch fabricating capability of the silicon technology and make hundreds and thousands of these electrodes with high reliability, with very efficient interconnectivity. And let's do that with silicon technology. So people switched to this silicon technology. The first electrode that came out was a Michigan microelectrode array from Kensal Weiss's lab. And all you're seeing here is tines of silicon with multiple recording sites on them that are implanted in the brain. So each tine would be about 50 micron wide, and or tens of microns wide. It depends. The design changes, variations are quite large. And they're several millimeters tall. And you got multiple versions of them. They're probably about a few tens of microns thick as well. So you're really talking about a very, very tiny structure. And this is a variation of that Utah electrode array. The problem is you're looking at a very complex space. I talked about neurons all the time. Now, but there's more than neurons in the brain. You got vasculature. You wonder, well, how many vasculature can you really have? Well, this picture captures some of the complexity of the vasculature. So if you look at the left picture here, I'm showing you a cube of a mouse brain, about 1.5 millimeter by 1.2 millimeter by 1.5 millimeter tall. So it's a pretty tiny cube of brain tissue from a mouse brain. And only the vasculature in that part of the brain. You can see the complexity of the vasculature. You see the large vessels on the top, those are the veins that run on the top of the superficial veins. And the big arterial vessels actually go way at the bottom of the brain, and they go up. The superficial veins go down, and you get a nice capillary bed that's visible here in this picture. So I had a movie on the right that shows when you poke these things with these electrodes that I showed in the previous slide, what really happens. So as you can imagine, if you take a sharp electrode or a sharp one of these needle-looking electrodes and you stick it in the brain, you're going to really rupture a bunch of these vasculatures. So what happens if you rupture these vasculatures? Well, you'll have bleeding. You'll have local bleeding. So what happens when you have local bleeding? You have stroke and a whole bunch of immune response and lots of bad sequelae happen as a consequence of that. How serious is this? You're going to have local bleeding. It's not too bad. So people have seen that. There's been extensive studies over the last 30 years plus where people have seen that these are manageable. You can still get meaningful readout from the brain despite all these disadvantages. So as I said, you rupture blood vessels, you create a lot of tissue damage. How does it look? This is a picture that captures some of that, where you're actually running a needle or one of these electrodes into the screen. So I'm showing you 3 different pictures of the same electrode. The brain tissue around it is characterized for inflammatory response. So there's a lot of all this green stuff and the red stuff and the blue stuff that you see here. The green and the red stuff is bad. The green is what's called the glial fibrillary acidic protein, which is a protein that's a marker of inflammation in the astrocytic cells. When the astrocytes are reactive, and they are reactive when they are in an injured state or a stressed state, they express this protein called glial fibrillary acidic protein. That's bad. You don't want to see that. And you also see the ED1, which shows the activated microglia. So that's also bad. So these are situations you have to manage. So what's the big deal? What happens if the tissue is damaged as a result of putting this? Does it prevent my ability to read these neurons? Turns out it does. Now then you ask a follow-up question, how bad is it? Can we manage this? Are there design can we design against it? Can you design to overcome it? So this is where the science of implants come in. So I'm going to talk a little bit about that in the next few slides. So in addition to these astrocytes that react and also the microglia that get activated, it turns out what happens is the neurons migrate away from the site. So the very neurons that you're actually trying to read from, they leave the site because of all this injury. So this is a picture that shows electrodes implanted in a cat brain. This is looking at it from the top. This is the brain and the vasculature here. And this graph here, there are 2 graphs. The graph on the left with a bar chart that shows as a function of distance from the center of the electrode, the number of neurons on the y-axis. So right up against near the electrode, when you're 0, you're up very close to the electrode, very few neurons. And as you migrate away from the electrode, 30, 60, 100, 80 microns away, you get more and more neurons. The neurons are either migrating away or they're probably dying next to the electrode, both of which are bad. This is another way of capturing the same information. Well, actually, this is another piece of information which actually says the number of electrodes that are actually functional after as a function of the days of implantation. So when you leave these electrodes in the brain, you actually have electrodes slowly fading away. So that's not good, because your goal, remember, is the three legs of the stool is to monitor as many neurons as you can, as long as you can, and in awake behaving animals. These electrodes allow you to monitor from awake behaving animals. That is not possible using an fMRI or some other cumbersome imaging technology. But the problem is they don't allow you to record for as long as possible. After a few hundred days, they start fading away. In fact, they actually fade away even earlier than that. These are from McCreary, Doug McCreary from Huntington Medical Research Institute. He's a highly skilled neurophysiologist. In his hands, these things are able to last as long as they do. With unskilled hands, the variability in the yield is even worse. So the reliability of this interface is very poor. So how reliably can I actually record from single neurons over time is very, very poor. All right, so that brings up the challenge of what are the challenges in being able to read from these neurons, and what are the challenges being able to write. So I kind of summarize all these different difficulties that I put up out there. You can build as much through capacity as you want. You can put as many recording channels as you want, or as many wires as you want in the brain, but what really matters is throughput. How much can you actually get out of this whole thing? How many are actually functional at the end of the day? And that's very poor. You can build thousands of channels, very easy. The technology exists right now for us to be building 10,000 channel electrodes. But when you stick them in the brain, not all of them are next to a neuron. So the yield becomes poor. And then not all of them last through the length of the time you desire. If you want them to be there for the length of the life of a patient, that they're not going to be around as long as you want them to be. The reliability is very poor. So that's the second point as well. Stability is poor. The signals, as I said, are a few microvolts to tens of microvolts to hundreds of microvolts. So that's the range of signals you're really looking at. And so there's a lot of non-stationarities, and the stability is also poor. The yield is poor. You put 100 electrodes in, you might get anywhere from 20 to 80 electrodes actually reading a neuronal population. And then this biofouling, the sensors get fouled up. All right. So what is our approach to this? What is the solution to this? So we have used 3 different solutions. One is automation in our lab, and the second is brain-like interfaces. And there's a fancy word for that called the biomimetic principles. And then we've also engineered some thin films at the level of materials. Can we redesign these materials to actually allow us to get these signals with high fidelity? So let me talk a little bit about all these 3 ideas, those 3 approaches. And before I launch into that, I want to talk a little bit about the science of these interfaces. As I said, there's a lot of inflammation that happens. There's a lot of the body mounts an aggressive immune response to this foreign body that's implanted. What do we know about this? What's the science of this? Do we know enough to be able to get around this? So one of the first things we did as we came into the lab, there was an existing understanding at that time that no matter what electrode you put in, as long as it's a foreign body, you're going to have a immune response from the brain. And that immune response gives rise to all the bad stuff that I mentioned earlier, which is there's this reactive astrocytes, this activated microglia, and the neuronal migration. All of this is bad stuff. So that was a prevailing understanding at the time. So what we asked ourselves was a very simple question. We asked, can we reduce the size of the implant, and will that reduce the inflammatory response or the immune response mounted by the brain? And the answer at that time was, generally, the opinion at that time was, well, it doesn't really matter what size you put in. So we tried a very simple experiment. We just put in an electrode, basically got a wire that was 12 microns in diameter, and another which was 25 micron in diameter. And turns out, we tried to put them in the brain and looked at the tissue response mounted by the brain to these 2 distinct size electrodes, the wires. It's hard to get if you actually handle a 12 micron diameter electrode, it's thinner, much thinner than a human hair. So it actually floats in the air. If you hold it up with one end, it actually floats in the air. It floats in the breeze. So it's very hard to poke it inside the brain. The brain is more like has a buttery consistency. So I'm just trying to explain this so that you get an idea of the mechanics of this whole problem. Turns out the mechanics is the key. So that's why I want to make sure you understand this. So that you have an electrode that floats around, you have to get it inside a brain that's buttery. So we had to use some kind of biodegradable polymer to coat it, and then you stuck it in. Turns out we got a surprising result, which is that what I'm showing you here is the tissue response on the y-axis, and on the x-axis is the size of the electrode. So this is 12 micron electrode after 2 days, 25 micron electrode after 2 days, not much difference. But if you leave those electrodes for 4 weeks, the 12 micron electrode is significantly less tissue response than the 25 micron electrode. So it is size dependent. So which is interesting. So the tissue response mounted by the body actually pays attention to the size. There are certain sizes that it actually ignores, or it does not really care much about. All right, so that's one of the pieces of the puzzle. So we call it the small is good idea. Okay, so our lab was the first to show this. Subsequently, Darrell Kipke from University of Michigan showed this as well a few months later. But this idea is now pursued widely as a way to actually design for minimizing tissue response in the implants that we put in the brain. The other idea here is regarding mechanical properties of the implants. So this is a fairly complex slide. I just want to quickly go over this is your biology, this is your material. All these things are the materials side of the things. What you have here is elastic modulus going from the brain that's here in a few kilopascals, depending on which paper you read and which lab reports this data. It can be anywhere between 500 pascals to 3 to 5 kilopascals. But the materials we put in are in the order of hundreds of gigapascals. That's your elastic modulus. So for instance, silicon is 100 to 180 gigapascal. Most metals are in that range. So you're actually putting a very rigid implant in the brain, which is very soft, and that could be a reason for this mechanical mismatch, is another reason for why this body mounts this aggressive immune response. So we made a material that's brain-like in its viscosity, and we showed that actually, so how do we know the viscosity of the brain? So we actually had to do some experiments to figure out, we had to develop a new technique called a penetrating microindentation technique. And then using that technique, we figured out we have an interface. So this picture here, this cartoon shows the electrode implanted in the brain, and all this stuff that goes on around the electrode is the scar tissue, or this tissue response mounted by the brain against this electrode. So clearly the mechanics changes. So we actually used this for 8 weeks. We put these electrodes in, we measured the mechanical properties. Up until then, people actually had biological characterization, but nobody had numbers to it. Nobody had measurements of what is the mechanical property of the brain. So we went and characterized the mechanical property of the brain. So the elastic modulus actually increases, of the interface increases and then decreases after 8 weeks, which is interesting. So we made a material like the brain, and it turns out actually when you take care of the mechanics, when you reduce the mechanical mismatch, the implant then is far more stable. So this picture here on the bottom right, I will not talk about the rest of the pictures in the interest of time. Bottom right shows you the impedance of this electrode, the electrical impedance of this electrode lasting for more than a year, 450 days. This is what we call a Nyquist plot. And all these different traces show when they lie on top of each other, it shows that they're very stable. The impedance is very stable. These two traces show electrodes without this coating, without this brain-like coating. These 4 electrodes show electrodes with the coating. They're far more stable. So soft is good. So that's the second design principle. The first design principle, small is good. So how small should it be? Smaller than 10 micron diameter is good. So small is good, soft is good. All right, so those are 2 key design principles. So we're going to use those 2 key design principles in our lab. We've used them extensively in our approach. Now, the rest of this presentation, I'm going to just walk through. It's going to be a walk through the museum. I'm just going to show you point my fingers at a few technologies that we've developed. So it's going to be very engineering at this point. So the first approach, as I mentioned earlier, of the 3 approaches, the first approach is the automation. Second approach is the approach using biomimetics or soft interfaces or brain-like interfaces. And the third approach is thin films. So this is the first of the 3, the automation. The idea here is that if you remember, I told you that we get poor yield. If you put 100 electrodes, only 20 to 80 electrodes actually record, or next to a neuron actually record activity. So there's poor yield problem, there's a reliability problem, and there's a signal quality problem. There's all kinds of problems there. So what we came up with a simple idea, an engineering idea, is can we put a motor on every of electrode here and move this electrode so that it's best positioned. Every electrode is independently positioned. Let's say you put 100 electrodes and you put a motor for each electrode. Can you move each of these electrodes independently so that they're right next to a neuron and getting the best possible signal? And so you get 100% yield. Not only that, when the time goes on, in case your electrode fails, your neuron migrates away or neuron dies, you activate the motor and go to a new neuron. So you always get 100% yield. So that's the idea of this microscale automation here. So the picture here on the left shows a slice of the brain, cortical brain with the pyramidal neurons. It's actually pyramidal neuron slices. And this is your robot. It's actually a cartoon. It's not the real robot. That's a control scheme. You record signals, detect these action potentials, see if it's good enough. If you like it, if you don't like it, you seek a new neuron. If you like it, you stay there. If you don't like it, you move the motor, go seek a new neuron, and keep doing that in a cycle. So that's your control algorithm here. So this, what I'm showing you in this slide is a product of two PhD dissertations, 3 different technologies, electrostatic home drives on the left, electrothermal actuators in the middle, and a 3rd generation of electrothermal linear ratcheting macroactuators on the right side. Bottom line here, I just want to get you to look at is there are many ways you can actuate these things, build these motors. How small are these motors? What I'm showing here, for instance, this is 600 microns in diameter. I'm sorry, the width is 600 microns. This is your electrode here, which is about 50 micron wide. And these are your gears that actually drive these electrodes. Your brain would be in the bottom here. So you place it on the skull, and these electrodes will actually go inside the brain, right next to the neuron you want to be recording from. So that kind of scale translates through all 3 of these different generations. So we've improved these generations of actuators. We collaborated with the Sandia National Labs in Albuquerque, New Mexico. The form factor for these motors are about 0.5 millimeter wide, mostly consistent through all these 3 different generations. Displacement resolution, they can actually move in resolutions of 5 to 10 microns. They can travel as much as 5 to 10 millimeters. So these are the electrodes, by the way, here on this picture, and this is the electrode here in the middle. And the motors then turn it and move these electrodes down in. So there's a lot of surface micromachining technology that goes in, which I won't get into. You need to be able to generate about 100 to 500 micronewtons of force to be able to push an electrode inside the brain. All right, so let me, at this point, switch to a movie here. I want to show you one movie that captures some of these. So what you should see here in a little bit is here you have a microchip. That's about, in this case, I have 2 electrodes. This is one electrode, and this is a second electrode. I'm driving the second electrode in. You're seeing it about, this is about 2 millimeters wide, and the electrode is about 50 micron wide. And these are the gears. You should probably be able to see the gears rotating. And the microchip is actually driving this electrode in steps of 10 microns. All right. So the whole electrode is about 7 to 9 millimeters long. So this is the end of the electrode that's coming in. So once it's in the right place, it will start recording. So this is all automated. So typically when I get a student in my lab and I have them do these experiments, if they have to use a wire by hand, I have to train them for several months before they actually get them right and they actually get the good data. Potentially with this controlled closed loop system with a motor like this, it should be plug and play. People should be able to go and put it in and start recording brain activity. Anyway, so how does this look like in the real world? So if you put this thing in the brain, after 14 weeks, this is how it looks like. It's pretty ugly. So you got to pay a lot of attention to packaging. And so we spent, believe it or not, about 7 years on packaging, because you cannot get something to work with so much junk in this microchip. So what you have here is a lot of blood, a lot of dry blood, a lot of cerebrospinal fluid, salt, and all kinds of skin exudates that have gone inside the chip. And remember, this is a microscale, micromechanical system. With all this junk there, it's not going to move. It's catastrophic. So it's not going to work if your packaging is not done right. So the packaging is a showstopper. You got to get that right. So it took us a good 7 years to get the packaging right for this microchip technology. And you have to package it in a way that is scalable. and it actually scales up to hundreds and thousands of electrodes. And you got to package it in a way that it's only chip size. So for instance, I'll give you a small flavor of the problem here. Conventional packaging would involve putting this chip on a chip carrier and on a PC board. By the time, so this chip actually has only 3 electrodes, but this is a microscale chip now became a millimeter scale technology. This is not a scalable technology. So we had to come up with things where you can actually stack these chips one on top of each other. So here I'm showing 3 different chips, each with 3 electrodes stacked on top of each other. These kinds of technologies we use a flip chip technology for this. We have 2 patents on this. I don't want to get into it because it's just too. All I want to help you understand here is that you got to get those small details, what looks like boring things right in order to get a technology right and functional. So this packaging is super critical. So the second idea I want to talk about next to automation is the biomimetics, which is when you make brain-like electrodes, they last longer. So we already proved that when the electrodes and the interfaces are brain-like and the material properties, the impedance is very stable for more than a year. There's another cool thing you can actually do with this. When we make this out of siloxane, siloxane is permeable to oxygen. Remember I told you these electrodes actually break vasculature as you go in? So it turns out, not surprisingly, the neurons in the vicinity of these implants have oxidative stress. So it's very crucial that you actually monitor the oxygen concentration, the partial pressure of oxygen in the brain tissue right next to these interfaces. So here's putting all these ideas together. We have a collaborator, Vikram Kodibakar, who actually has a technique by which he can measure oxygen. He calls the technique pistol. He's using an MR imaging technique. So we got together and said, hey, can I use my siloxane-based brain-like interface and measure oxygen using your technique? So those 2 minds got together. When you have it, now we have a technology by which we have an electrode array made of siloxanes. You stick it in the brain. The oxygen in the vicinity of the brain comes. It's a hydrophobic material, the siloxane. And again, the scale of these electrodes, the tines here are about 150 micron diameter, each of these tines. It's oxygen permeable. Comes in, it changes the relaxation time constant of these siloxane molecules. So under external MR magnetic field, you can actually pick up the change in the relaxation time constant, which is a function of the partial pressure of oxygen. So you can actually get a readout of the oxygen along the length of the shank while you're measuring the activity of the neuron. And this is really very interesting because now you can get metabolic, the effect of the metabolism of the extracellular space, the oxygen demands, and how it affects the neuronal function. So this is funded by the NIH Brain Initiative. I'm going to skip a few slides here in the interest of time. There's thin film technologies here. This is interesting because we found out that, interestingly enough, we work with polysilicon, machining those, making those interfaces that I talked about. And the polysilicon is polycrystalline silicon with many grain boundaries. And it turns out you can actually suppress noise by just using a polycrystalline material because the IV characteristics, the current voltage characteristics, you have current voltage characteristics here is non-linear for a polycrystalline silicon. It's not like the wire, which is linear. And turns out the nonlinearity occurs exactly at the voltage corresponding to the noise. So bottom line, forget about all these equations on the slide. When I stick these polysilicon material in the brain, I have neuronal activity and I have biological noise that gets transduced through this polysilicon material. When the noise, which is less than typically 25 microns, looks at this polysilicon material, it looks at a high impedance pathway. When the neuronal activity, which is typically more than 25 microvolts, it looks at this polysilicon material, it sees a low impedance pathway. So interestingly enough, that material by itself acts like an in situ noise suppressing system. Very interesting. Passive, no batteries, nothing required there. So that's one idea. Second thin film idea I want to lay here is you've made organic LEDs by which you can actually use it to excite neurons. What's the big deal about organic LEDs? The organic LEDs are, you can make them on plastic substrates, so they're flexible. Remember, I told you, if you can make it brain-like, it's good. So if you can make it flexible, that's good as well. So you can make them on plastic substrates that are flexible. You can also reduce the interconnects. Interconnects are so crucial because if you have, let's say, 100 by 100 array of LEDs, you'll need 100 times 100 wires to activate each LED. So that's 10,000 wires. Using an organic LED, and with an addressing technology like the one I show here on the top right, you can actually do 100 plus 100 with 200 interconnects. So you just, in one shot, reduced the number of wires from 10,000 to 200. So the organic LEDs helps us minimize interconnects, low power operation, and also allows us to excite neurons. So we proved that, we published it recently, and you get some good looking neuronal activity with this optical. You may ask, how can you actually excite neurons with light? It turns out you can do that through what is called optogenetics. So you actually make the neurons express an opsin, which is a protein. It's a transmembrane protein. You make the neurons express that in the first place by transgenic. You actually put a plasmid inside these neurons. So they genetically express an opsin that is sensitive to light. So when you shine a light of a certain wavelength, it opens up, the pore opens up and allows, generally it's non-specific to what kind of ion it comes in, but they depolarize and they start firing action potentials, depending on the opsin. You can also have an opsin that actually inhibits the cell. So we've tried that as well. So you can actually do all these excitation, inhibition, all that is possible. Last couple of ideas. One is for injectable neuromodulation. We have a technology that we're working on that allows us to inject small microscale particles into nerves that you can then activate using sound waves from outside. So this is a two-piece technology. So why would you want to do that, and why is that necessary? So here is a picture of a nerve fiber. So this is not the brain, but this is actually a nerve fiber in the peripheral nervous system. So if you take imagine a nerve fiber going into the screen, the picture here is a cross-section of that nerve. And what it shows you is a cable, a bundle of bundles. That's kind of how it looks like. If you take the sciatic nerve in your leg, for instance, you slice it, you'll see thousands of axons like cables running down, and they're a bundle of bundles. So there's one bundle here, one fascicle, another fascicle, another fascicle, each with bunch of axons in them. And these white stuff that you see here is actually the electrode array that's implanted. So they're actually poking in, the needles poking in. So ideally, you want to be able to access individual axons. So what we are proposing here, and what we have done and we've tried here, is to put injectable particles, very microscale particles. If you can get them in here, and the particles don't have wires. So the problem here is that in the peripheral nervous system, particularly in your hands and your legs and elsewhere, the wires can lead to great instability. So a wireless and a batteryless technology would be highly preferable. I'll just lay the principle down here. It's a two-piece technology. It receives sound. You shoot sound waves through the skin, and this particle inside the body, which has a piezoelectric material, receives the sound waves, converts that to electricity, and it has a local diode in place that converts the sine waves to rectified sine waves. Interestingly, the neurons don't respond to sine waves, but they respond to rectified sine waves. Neurons respond to sine waves at a high amplitude, even the frequency is very high. So you can actually rectify them locally and stimulate the neuron of interest. So that's the concept, and this is our implementation. So we show here a sciatic nerve in a rat. This white thing is your sciatic nerve. And the black powder that you see here is really the particles. It's a two-piece particle again. One, it has a diode and a piezoelectric material. These wires are really a cuff electrode. They don't belong. We're using that to verify if these wireless, batteryless pieces are exciting the neuron the same way a wired cuff does it. So that's an experimental data here. All right, let me conclude here and then talk for a few minutes, talk about a couple of other things. Automated systems can achieve high yield. They can be adaptive to structural and functional changes in the neural circuitry and the brain environment. Stability can be enhanced using automated systems. Signal to noise ratios and interconnect issues can be significantly improved using thin film technologies. High information throughput can be accomplished by building high density arrays of movable electrodes. Multifunctionality, the kind of electrodes I talked about, we're doing oxygen measurements, and electrical activity at the same time, gives us a potential to discover mechanisms of how metabolic demands impact functionality of neurons. So in the last couple of minutes or so, I want to talk a little bit about having a successful engineering career. The question I asked myself here is, what one thing or a few things that I wish somebody had told me when I was in college, when I was in grad school? So I just listed a few of them, not all of them, but a few of them here. The first one is entrepreneurial mindset. And I'll talk a little bit about that in the next slide, which is my last slide. The entrepreneurial mindset is identifying a problem has a 4-step process here. Identify a problem, generate a concept to solve the problem, pick the concept you think best meets that problem here, and then go into prototype development. Those are 4-step process, and I'll talk a little bit about that more in the next slide. But 2 other ideas and 2 other things I wish somebody had told me when I was in college. Those are very simple things. Write every day. Write a little bit every day, no matter what stage in your career you are, because writing is equal to thinking. It actually clarifies your mind and actually helps you generate new ideas. So write every day. The last thing I would say is read some books that are outside engineering, philosophy, history of science, and things like that. They really stretch your mind. And all of the tips that I'm giving you here are very simple, but they're not necessarily easy to do because they involve some discipline. So regarding the entrepreneurial mindset, I'll give you 2 examples, 2 stories. I'll end the talk there. The first story is my class, which is called the BME 370, Biomedical Engineering 370, which is 3 stands for the junior level class, which is 3rd year in the engineering program. This is a class called Microcomputer Applications in Bioengineering. What I do in this class is very hands-on, project-oriented class, and I ask the students, give the students a lot of skills. I teach them a lot of skills. So that's one part of the class. A second part of the class is mindset development. So one part of the class is a skill set development. The second half is a second part of that, not half. Second part is a mindset development. And that's the entrepreneurial mindset. So I have the students, I send them out for 3 weeks in groups of 3, and I have them go and find a problem. And this is what I want to leave you guys with. Every one of you who's listening to me, find a problem that you really want to solve. And you don't really need to have all the skill sets to solve the problem. Generally, when I ask students, what do you want to do? The mindset, and I was in that same mindset as well. So I want to get a job, or I want to go for study this or study that. I'm suggesting to you an alternate way of thinking about it, which is solve a problem. Find a problem and solve a problem. And that problem could be anything of interest to you. And you will find very soon that finding a problem, even finding a problem, is not that easy. You got to pay attention. Many things happen. There's lots of things you overlook in life. But identifying a problem is not easy. But if you can find one, look for a problem that you can solve. It doesn't matter if you're a freshman, you're a 1st year, 2nd year, 3rd year, 4th year. It doesn't matter. It doesn't matter how much skills you have. It doesn't matter what grades you get. Every human being can solve a problem. Every human being can identify a problem. So I would strongly encourage you all to find a problem that you can solve. Make that your passion. Make that your pain point. Make that your pain. And then try to solve the problem. You'll find that the world gives you a lot of opportunities to solve it, no matter what you choose to do in your future. Whether you want to do grad school, whether you want to go and work in a company, have a problem that's your own passion. This is a story of a company called Neolite. I had a student by name Siva Kumar, who joined me from Chennai many years ago. And he did masters with me in our lab. He founded a company called Neolite, which is quite unusual for a student from India to do. And this company actually makes phototherapy devices for infants with jaundice. Infants with jaundice is fairly common, and phototherapy is also fairly common. All he made was a fairly simple idea of shining light using LED on these infants as a therapeutic device. And this company is taken off. Huge success. And it's got FDA approval. It's got lots of money. He raised a lot of money. And he's moved on in life. So he's still around. But I just wanted to share the story with you. Is something that unusual that most students from India don't necessarily think of those possibilities. There's so many problems around us. You just pick the one that you like. You'll find that there's lots of ways to people to assist you, lots of resources to assist you as well. And I want to thank my students. This is my team of students and my collaborator, Dr. Bruce Stowe, with whom I've had years of very intense, close collaboration. Other collaborators include Murad Okandan from Sandia National Labs, Michael Baker as well, Jeff Capadona from Case Western, Joe Pancrazio from UT Dallas, and Eric Maas from Medtronic, and a bunch of other people. And students, all of these people, and funding from federal agencies, NIH, DARPA, Arizona Biomedical Research Commission, and Whitaker Foundation, all of which, without whom, none of which should be possible. And I'm happy to take any questions. Thank you.
Sir?
Yes.
So the first question is, what are the side effects of deep brain stimulation?
That's a good question. Depends on where it's implanted and what kind of dose of stimulation you're giving. And the side effects, I have actually a student in my lab who has this thing implanted in his brain. People have, for instance, people with Parkinson's disease who have this deep brain stimulation can potentially have, if it's not in the right place, if it's a little bit in the wrong place, you can have anywhere between slurring of speech to sometimes cognitive deficits. And my student, for instance, he wears this and he goes on the treadmill. And interestingly enough, as he goes on the treadmill, his mouth starts to feel, he has some weird sensations in the mouth. So he actually turns it off and then does the treadmill. So as he bounces on the treadmill, the stimulation does some things to his mouth. So the side effects so far for Parkinsonian disease, where the target is well identified, the side effects are not as severe. But for other disease states, like mental health disease states, like OCDs or treatment-resistant depression, where the targets are not identified yet, we don't know which part of the brain to be stimulated for maximal effect. There, the side effect is a little question. It's a work in progress. So it really depends on the disease state, where it's implanted, and how well it was implanted. But as far as Parkinson's disease goes, the side effects are not that severe.
So the second question is, can you elaborate on brain chip?
On the brain chip?
Yeah.
So exactly what do you want me to talk about the brain chip? Is it clinical, or what part of it do you want to elaborate on?
The clinical part, sir.
Okay, so let me take a guess and as to what the intent of the question here is. So can it be translational? The answer is yes, it can be translational. There are challenges being translational. Anytime you have wires and leads going into the brain, there's huge challenges. So let me rephrase the question into saying, what are the translational challenges in getting this brain chip into a human patient, for instance? It's all cool to make microscale devices, but you don't see many microscale devices actually make it to the clinic. Eventually, what makes it to the clinic is what can be handled successfully by the surgeons, neurosurgeons. If I give one of my chips to the neurosurgeon, he'll break it. He won't be able to handle it. It's not ready for translation unless we deal with some of those issues. It needs to be robust. The packaging needs to be robust. It doesn't mean it's impossible, but those are some things you want to worry about. You would certainly want to eliminate leads as much as possible. So wireless technologies are crucial. They give you a lot of advantages over wired technologies, but the wireless technologies have a battery life associated with it. So if you can somehow find a way to communicate or transfer energy into the body, especially into deep structures, very efficiently, I think you have a winner. We chose sound as a way to transfer energy. People have tried RF waves, but that's too lossy, too many losses. Light doesn't get in there. If there's some other way to get energy into the brain in deep brain structures, if we're talking about multiple centimeters into the brain, we have a winner. So translational challenges, wires, wireless technologies, batteries, lead, mechanical issues with leads, leads pull. When you move around, the leads tug at you, and then they get out of place. So all of a sudden, you're stimulating a wrong place in the brain. So those are all issues there. I hope I.
Yeah, sir. Yes, sir. And the next question is, what is the success rate of the spinal cord stimulator?
Ooh. Okay, that's a difficult question because the spinal depends again on what you're using it for. If you're using it for pain, there's a company called Nevro that, again, what product are you using and where exactly are you implanting and what disease, what kind of pain are you trying to address? So there's lots of questions. Nevro uses a different kind of technology that's a high frequency stimulation. So if you, there's a range of numbers here. So if you're trying to address pain, you can put it in the dorsal root ganglia, for instance. It's just been a very interesting location to put it in, or you can actually put it elsewhere on the spinal cord. So it depends on where you put it in. Your numbers could be anywhere between 30% to 70% success rate in terms of finding some improvement and mitigating pain. What kind of pain? We're talking about lower back pain, usually, if you're putting it in the spinal cord. People after surgery, it's called failed back FBS, failed back surgery pain or something like that, where people with back surgeries end up with a lot of pain. It's very debilitating. You lose sleep. You can't do much of the things that you need to be doing. But the success rate, if you want to call it success rate in terms of mitigating pain, it can be anywhere between 30 to 70 or 80%. It's a wide range.
Okay, sir. And the next question is, are the brain implants the future of thinking?
Okay, so you're getting into dangerous territory here. There is a program from DARPA which is actively pursuing that ability to read and write into the brain. And yes, so the short answer is yes. Do I want to do it? If for some reasons, if there is a disease state or you want to, as a therapeutic modality, I'm all for it. But if you want to put it in healthy people and want to read people's brains and write into people's brains, which is what the defense wants to do it, I'm not there. So it depends really on personal preferences. But yes, the technology is going there. You will see if not in a few years, I imagine probably in the next 3 to 5 years, you should see technologies that come where you're actually using them in humans to read brain activity and write into their brain as well. When I say write, it means controlling the brain. I don't know if any of you would volunteer to do that, to be controlled by somebody else.
Yes, sir. And the next question is, what is the lifetime of an implanted material?
Yes, that's a good question. Depends a lot on the dimensions of the implant. So as I was tried to stress in my talk, is the mechanics is very important, which is very interesting. It's not the way normally we think about it, right? It's everything is electrical here, and there's biology, then there's electricity. But turns out if you take care of the mechanics, then the electricity takes care of itself. So if you get the mechanics right, small enough, soft enough, flexible enough, whatever, then you can actually make these things last for several years in the brain. People have had it in monkeys, non-human primates, for 7 years. So this can be made to last for several years if you do it right.
Sir, and the next question is, what about the biocompatibility of an electrode?
So it's a very related question. So all those tissue damage pictures that I showed address the issue of biocompatibility. So there is a 1960 paper by Stensas and Stensas that actually gives you a laundry list of all the different materials that are biocompatible and that are not biocompatible. And so there's some materials that are good for the brain, some are not. So you pick from that list, and then you work through the mechanics of those materials, making them the right shape and size and all that. Then you can minimize the tissue response. You can't eliminate it completely. But if you get it down to really tiny sizes, like what we have seen from Charlie Lieber's lab in Harvard, if you can get them down to less than 10 microns, perhaps even smaller, he has got them down to cell scale. And he's seeing almost no tissue response, no immune response, so perfectly biocompatible according to him. So if you can engineer it down to that size, yes, you can. It's possible. So you can make it perfectly biocompatible in the brain. You don't need to get that far, but yes, you can do it. It's possible.
Sir, and the next question is, what are the type of materials which have been used in an electrode?
Okay, so generally want to use materials like the word that you want to use here. I don't want to use that word because it might lead to some confusion. It's a very inappropriately worded. There are two types of materials: polarizable or non-polarizable materials. So let me not use that terms because it leads to confusion more than clarity. Materials for electrodes: gold. We have used conductive polysilicon. As I said, in the thin films, it actually gives us very high signal-to-noise ratio because of the suppression of noise. Platinum, iridium oxide films, and interestingly enough, some conductive polymers as well. Some polymers are conductive, like PE-DOT is a conductive polymer. Polypyrrole is a conductive polymer. So people have used conductive polymers like PE-DOTs as well for achieving electrodes. There's some advantages to PE-DOTs. The traces can be anything. You can use all kinds of traces because you're going to insulate them, but the electrodes themselves could be any one of these materials. And the bulk on which you lay down these electrodes, the bulk material, the shank or the tine or the needle, that can be silicon. If you're using a silicon-based technology, it could be other insulator of choice.
So the next question is, what are the possible drawbacks of the implant?
Possible drawbacks in terms of functionality?
Yeah, sir.
Okay, so patients have used these implants. I showed you some pictures of patients actually having this in the motor cortex. They have had hundreds of electrodes in the motor cortex for years. And they are able to control robotic limbs, for instance. They are actually able to feed themselves, they're able to do things and so on. So yes, it's possible to have them without any, if you can do the surgery clean, if you have a good neurosurgeon, there's really no reason to worry about bad consequences. But there's all kinds of advantages to be had from using these kinds of things. So in terms of bad, I'm not sure if the question was necessarily worded for bad consequences or infections and things like that, which is a huge risk, by the way. You can, if you do it right, you can avoid all those infections and so on.
So then the last question is it possible to visualize the communication with the brain?
Visualize the communication. You can visualize the effect of the signal you're putting into the brain. So if I'm stimulating the deep brain structures like the subthalamic nucleus, I can see the tremor stop. So I can visualize the effect of stimulation. I can see the signals coming out of the brain on a computer screen because it's electrical activity. It's like any other electrical activity. Unless I'm imaging calcium activity or imaging some other functional marker, visualizing brain function is you can visualize the electrical activity and put it on in a picture. That's about all you can do.
Yeah, sir. Thank you, sir, so for patiently answering all the questions which has been addressed by the participants.
Thank you very much for this privilege. And thank you for all your questions. Please feel free to email me. Look up this website that I have on the screen if you have further questions or if you want to read up on some of my papers. And if you have any questions, you're welcome to direct them either through Bhaskar or you can actually email them to me at jit@asu.edu. That's my email. That's jit@asu.edu. jit@asu.edu.
Okay, thanks for sparing your valuable time with us, sir. So more than 400 participants attended your lecture today, sir. So I want to conclude this session with vote of thanks. I would like to express our sincere thanks to our guest, Dr. Jitendran Muthuswamy, for sharing his valuable ideas in this international lecture. I would also like to thank our beloved HODs for their moral support and guidance. I'm very happy to express my vote of thanks to our staff members for their valuable suggestions and participants who have made this lecture a great success. Thank you. Thank you all.
Thank you.